Hello viewers, welcome to the Eastern Cape Department of Education's broadcast studio. I am Mrs. Nkosha from Richard Vacha Technical High School. I am a life sciences educator. Today we are going to be covering the topic genetics, and we will be looking at concepts in inheritance for your full understanding. For today's lesson, we are going to be using the examination guidelines as a lesson content or a guide for this particular lesson. Now, it is very important to use the exam guidelines whenever we are studying, because remember, in life sciences, we use a lot of different textbooks, we use a lot of different study guides. Sometimes they fall short of the information that a learner is supposed to have of what is examinable. Sometimes they go beyond the content that is examinable. So the exam guidelines, it guides us to exactly what it is that will be covered in the exam. So very importantly, whenever we are studying, we need to incorporate the exam guidelines, and each and every learner should make sure that they do have a copy of the exam guidelines. Now, the first concept that we are going to be looking at today, we are going to be talking about Gregor Mendel as the father of genetics. We are also going to be talking about chromatin and chromosomes. We are going to look at genes and alleles. We are going to look at dominant and recessive alleles. We are going to look at phenotype and genotype. We are also going to differentiate between homozygous and heterozygous alleles, and we are also going to be looking at Mendel's laws. First one will be the law of dominance, and the second one will be the principle of segregation, and we are also going to be looking at the basic format of a monohybrid cross. Now, what is genetics? Genetics is described as the study of heredity. This is how characteristics are passed on from parents to offspring. Now, if you look at this example over here, we can see here that we have got two parents and we have got their offspring. Now, remember, offspring receive half of their DNA from each parent. So it means that this child has received half of their DNA from the father and half of it from the mother. And if we can see now the characteristics that this child has inherited now, he has inherited characteristics both from the father and the mother. Now, if you look at the color of the hair of the child is the same as that of the father. But then now the shape of the hair the child has inherited from the mother. Now, if you also look at the nose, you also look at the eyes, you will see that different characteristics have been inherited differently in the child, because now they have received DNA from both the mother and the father. Now, if you look at this second picture, we have got here an adult male and an adult female. But when we look at them, the female looks like a little baby, and the male looks like he could be the father of this female. But actually, this female is 33 years old, but she is only 63 centimeters tall. The male is in his 40s, I think 42 to be exact, and he's currently the world's tallest man alive today. He is 42 and his height is about 2.72 meters. Now, with this picture, I want to show you that what an individual looks like, it is as a result of the DNA that they have inherited from the mother and the father. And here now in genetics, we are going to be unpacking how this happens. Okay, and then now over here on our far right, we have got Gregor Mendel. Gregor Mendel was an Austrian monk and also a biologist. He was the first person to study how characteristics are passed on from parents to offspring. Now, when Mendel was doing his experiments, or when Mendel was trying to explain how characteristics are inherited, he was looking at pea plants. Now, some of the characteristics that he looked at in pea plants, he was looking at the seed shape and color. He was also looking at the pod shape and color. He was also looking at the inheritance of flower position and color. And he was also looking at the inheritance of size of the plants. And from his experiments, he came up with three laws: the law of dominance, the law of segregation, and the law of independent assortment. And in this particular lesson, we are only going to be covering the first two laws. Now, if you look at this diagram here, that is on our right, we can see here that there are two plants here. There is a tall plant and there is a short plant. Now, pea plants are self-pollinating plants. So Mendel noticed that tall plants tend to produce tall offspring and short offspring tend to produce or rather, short parents tend to produce short offspring. But now, when he did a cross between a tall parent and a short plant, he noticed that all of the plants that are produced in the first generation are tall. But then now, if he cross-pollinates two of the plants that are tall from the first generation, he would notice that now the offspring now will give him a mix of tall plants and short plants as well. But the genotypes that you are seeing here, we are going to be talking about them as we go further in the lesson. Now, moving on to the important concepts. Now, remember, in life sciences, it is very important to make sure that for each and every topic, you fully understand the terms and the concepts, because they are going to give you the foundation of knowledge that you need in order to have a full understanding of the topic. So now in genetics, now one of the most important terms that a learner needs to understand is chromatin. Now, chromatin is not new to us because you have learned about chromatin when you were doing meiosis and DNA in term 1. Now, chromatin is defined as a network of DNA and proteins that forms chromosomes in the nucleus of eukaryotic cells. And chromosomes are defined as thread-like structures that make up DNA and protein that are found in the nucleus of most living cells carrying genetic information. Now, we know that if we look at this diagram here on the right, we know that DNA is found inside the nucleus. So initially, DNA is found in the form of chromatin, or this tangled up network of DNA. Now, this chromatin, it then condenses in order to be visible as chromosomes. We have learned this in term 1. Now, we know that during prophase, DNA condenses and becomes visible as chromosomes. So, very importantly, our DNA now as humans have got 46 chromosomes, as you already know. So it means that the entire human DNA is covered, or it is contained in the 46 chromosomes that are there in the nucleus. So this is very important now to understand, because as we are going to be talking about genes, genes are found inside these chromosomes. Now, how do we define a gene? We say that a gene is a small portion of DNA that codes for a particular characteristic. Here on our right, we have got the structure of a DNA molecule, and we know that DNA is a double helix structure. Now, there are segments or pieces or portions within this DNA that are called genes, and very importantly, these small portions, each of them codes for a particular characteristic. And in term 1, we also learned that some parts of DNA are non-coding DNA. So very importantly, a small portion of DNA that codes for a particular characteristic is called a gene. But now remember, how do we find this DNA in the nucleus? We find this DNA in the nucleus in the form of chromosomes. So now, we have got alleles. What are alleles now? Alleles are different forms of a gene which occur at the same locus on homologous chromosomes. Now, remember, when you look at a human karyotype, you will see that chromosomes now, they occur as pairs. We have got homologous chromosomes, and how we define them, we say that homologous chromosomes are a pair of chromosomes. One is from maternal origin, the other one is from paternal origin. And now the characteristics of these homologous chromosomes is that they have the same size, they have the same shape, and they have got the same position of alleles that code for the same genes. Now, if we look at this particular diagram, you can see that now this diagram has represented a single gene. what we need to know and what we need to remember is that a child does not inherit a single gene from a single parent, but rather they inherit an alternative form of a gene from one parent, which is an allele, and another alternative form for that gene. So it means that two alleles, one from each parent, they join and they form a single gene. So now offspring do not inherit a complete gene from each parent, but rather they inherit an allele from each parent. Now, let's take an example here. Before we go to the example, let us define genotype and phenotype so that we can clarify these alleles and explain how we use letters to represent these alleles. Now, the genotype is the genetic composition of an organism. So it means that this is the DNA that the organism has inherited. But then now what we see is the physical expression or the appearance of an organism, which we call now the phenotype. Let's look at an example here and look at how eye color is inherited. Now, we have been told here that the allele for blue eyes is B, and the alleles for brown eyes is capital letter or uppercase B. Now, question 1 is asking, what is the phenotype of picture A? Remember, we have just defined phenotype as the physical expression or the appearance of an organism. So now, if we look at diagram A, we can see that now in diagram A, we have got blue eyes. So it means that now the phenotype of picture A would be, the phenotype here would be blue eyes. And then the second question now is asking, what is the genotype? Remember now, the genotype is the genetic composition of an organism, and it shows us now the alleles that have been inherited. And remember, we were given an allele here for eye color, which is small letter B for blue eyes. So it means that now the genotype needs to show two alleles, because one allele is received from the mother and one allele is received from the father. So this genotype of having two small letter B's will give us now the phenotype of having blue eyes. Now, if we look now at the second diagram, or the second question is saying, what is the phenotype of picture B? Now, if we look at picture B, let us remind ourselves again that we said that now the phenotype is the physical expression or appearance. So now if we look at the second diagram, these eyes appear to be brown. And we were told that the allele for brown eyes is B. But now remember, a genotype now needs to show two alleles. So it means that now the genotype for this particular one, for now, we can represent it as two uppercase B's. Now, let us move on and talk more about these alleles. Now, on the right here, we have got a diagram that shows us more about alleles. So now the first thing that we are going to talk about in defining alleles more, we are going to talk about the locus. Now, the locus is the exact position or location of a gene on a chromosome. Now, remember, we had said that a single gene will have two alleles, one from each homologous chromosome. Now, if we look at this particular diagram, how many genes do we have here? We have got this gene, which has two alleles. We have got also the second gene, which also has two alleles. We have got the third gene with two alleles, the fourth gene, and the fifth gene. So it means that now from this particular diagram, we have got 5 genes. Because we had said that an organism does not inherit a gene from one parent, but rather inherits an allele from each parent. And now those alleles, when they are sitting there on homologous chromosomes, they form a single gene. Now let us go to the second definition. The second definition, it tells us about homozygous alleles. So homo, in simple English, homo means same. So now when you have got homozygous alleles, you have got two identical alleles for a particular characteristic. So from our diagram here, we can identify that gene number 5, this one, is homozygous because now it has got identical alleles. We can also identify that gene number 3 is also homozygous, but then we can see that the other ones have got 2 different letters, so it means that they are heterozygous. Hetero in simple English means different. So even now, if we are saying now we have got a heterozygous genotype, it means that it contains 2 different alleles for a particular characteristic. Now, if we go back now to our diagram, we can see that from this diagram, gene number 1 and gene number 2 and gene number 4 have got heterozygous alleles. Because now the alleles, you might be asking yourself, why are we representing these alleles as small case letters and as capital or uppercase letters? It is because some of these alleles are dominant and some of these alleles are recessive. So how do we define a dominant allele? Now, a dominant allele is an allele that is expressed in the phenotype when it is found in the heterozygous and in the homozygous condition. You have been given example here. The example that we have been given here is that if we have got an allele for being tall and we have got an allele for being short, and now this allele for tallness is present in its homozygous condition, meaning now we have got 2 of the same allele, and also in its heterozygous condition, meaning now we have got 2 different alleles. Now this particular one, it will always show in the phenotype whether you have got 2 of the same alleles or whether you have got an allele for tallness and an allele for shortness. So it means that tallness is dominant over shortness. When you see an individual that is tall, that individual can be homozygous tall or that individual can be heterozygous tall. For as long as that individual has inherited the dominant allele, it will be expressed in the phenotype. It does not matter if it is in its homozygous or in its heterozygous condition. Now, we go to the recessive allele. Now, the recessive allele is one that is masked in the phenotype when it is found in the heterozygous condition. So it means it is only expressed when it is in the homozygous condition. Now, from the example above, we have seen that if you have got 1 allele for tallness and 1 allele for shortness, this individual will be tall. Why? It is because this recessive allele is masked by the presence of the dominant allele in the heterozygous condition. It will only be expressed in its homozygous state, meaning we need to have 2 recessive alleles for it to be expressed in the phenotype. So an organism will only appear short when they have inherited 2 recessive alleles that are the same. Now, we need to look now at an activity so that we can see that we understand what we are talking about with these alleles. So our activity is saying the paired alleles in the diagram below represent a gene. A number of genes for different characteristics are shown. Write down the relevant letter A to D. For A, the homozygous state. Now remember, we said that homo means same. So it means that now we need to look for the letter that represents alleles that are the same for the gene. So now if we look at letter A, it is pointing at different alleles and alleles for different genes. Now if we look at letter B has got the same alleles for the same gene. So letter B, it represents the homozygous dominant state. Number B, write down the relevant letter that shows 2 alleles for different genes. Now again, we go back to A. Remember, how many genes do we have in this diagram? We have got this gene here that is at this locus. We have got a second gene here. We have got a third gene here. We have got a 4th gene. and we have got a 5th gene. So it means that now if you look at letter A is pointing at 2 alleles from different genes, whilst the other letters B, C, and D are pointing at alleles for the same gene. So the correct answer here would be A. Then now let's look at C. Now, which one represents the homozygous recessive state? Now, remember, what is homozygous? Homo means same, but then now it is being coupled by what? By recessive. Now, recessive, remember, we represent it in small letters. So it means that now we have got letter B has homozygous, but because it's uppercase letters, it means that is homozygous dominant. And then now if you look at letter C, it has got 2 alleles that are represented by small s letters. It means that this one now is the homozygous recessive state. So the correct answer here would have to be C. And then we look now at the last question. It is asking which one now is the heterozygous state. Now remember, we had said that hetero means different. So it means that now we are looking for 2 alleles that are different. How are they different? One will be an uppercase letter, the other one will be a smaller case letter. So now it leaves us with what? With D, because now it has got different alleles. So the correct answer here would be D. Now, let's move on. We have got a second activity here. We have got a table that has got a missing genotypes and phenotypes. But before we fill in the missing genotypes and phenotypes, let us see what is it that we have been given. Now, for the first one, also we have been told that these are the letters that we should use. Okay, now for the first one, it says homozygous. So it tells us that for the alleles, we are going to write the same alleles, but then now they are dominant. And now if we look at the phenotype, we can see short, which is a recessive phenotype, and then now, if now we have homozygous dominant, it means that the alleles now here have to be the same and they have to be dominant. Hence, we are going to have 2 capital letters, TT. And the phenotype now, this dominant allele now results in a phenotype that gives us a tall phenotype. Okay. Now, let's look now at the second row. Now, the second row, what are we missing here? We are missing the genotype, but what have we been given? We have been given short, and we have been given that the alleles for short, it's only small letter t's. And remember, we had said that now these are recessive, and we said that now the recessive alleles are only expressed in the phenotype when they are in their homozygous state. So it means that here we will have to have a homozygous recessive. Now I am overlapping here with my writing. So we have got homozygous recessive, which will give us a short phenotype, which the alleles we have already been given. Now I'll just remove this a bit so that I am able to write the third answer. Okay, now what have we been given here? Now the third one, we have been given now 2 different alleles. So now if we are given now 2 different alleles there, it means that now it has to be heterozygous. Okay, and now the heterozygous, it will give us a tall phenotype. Why is it giving us a tall phenotype? Because now for heterozygous, it means that we have got a dominant allele and we have got a recessive allele. And now in the heterozygous condition, it is only the dominant allele that gets to be expressed in the phenotype. Hence, our correct answer for the phenotype here is tall. Now, moving on, let us look at Mendel's laws. Now, remember, we said that Mendel is the father of genetics. It is very important to remember the names of the scientists because in the exam, they will ask you to associate certain scientists with what they have come up with in life sciences. For example, you have seen that in DNA, they were always asking you who were the scientists that are involved in the discovery of DNA and who were the ones that were involved in the discovery of the double helix structure of DNA. So it is the same here. When they ask you which scientist is the father of genetics, you need to know that it is Gregor Mendel. Now, let us look at Mendel's law of dominance. Mendel's law of dominance, what is it saying? It is saying that when two homozygous organisms with contrasting characteristics across all the individuals of the F1 generation will display the dominant trait. Now, what does it mean to have contrasting traits? Let's make an example. If now you have got an organism that is homozygous tall, that is crossed with an organism that is homozygous short, because tall and short are contrasting. So it means that now all the individuals of the F1 generation will display the dominant trait, which is what? Which is tall. Now, the second part of the law of dominance is saying an individual that is heterozygous for a particular characteristic will have the dominant trait as the phenotype. Now, let's look at this resulting individual here, because now they said that when two homozygous organisms, so it means we have got homozygous tall and homozygous short, it results now in heterozygous tall. And this heterozygous individual in the phenotype, they are displaying the genotype of the dominant one. So it means that now in an exam, when they ask the law of dominance, you can either write the first part, that when two homozygous organisms with contrasting traits are crossed, all the individuals of the F1 generation will display the dominant trait. Or you can write the second part that says an individual that is heterozygous for a particular characteristic will have the dominant trait as the phenotype, because they mean the same thing. Now, if we look at these diagrams that we have here as an example, let's first look at the one that is here on the left. If we look at the diagram that is here on the left, we have got here two different phenotypes. We have got cherries here, this fruit. The first one, we can see that this one is homozygous dominant and it is red. The second one is homozygous recessive and it is yellow. But then now the offspring now is displaying the phenotype of the parent that had the dominant alleles. So also both parts of the law apply here, because the first one is saying you have got two organisms with contrasting characteristics, as we have seen, and then all the individuals of the first generation produced have the dominant trait. And also, if you look at this second part of the law, that says an individual that is heterozygous for a particular characteristic, as we can see this one here, it has now the dominant trait as the phenotype. So this is the law of dominance. It means that whenever there is a dominant allele, it will always appear in the phenotype, whether it is in the heterozygous condition or in the homozygous condition. But remember, the recessive allele, it only appears in the homozygous condition. Now, let's also look at the second diagram here. The second diagram here, it shows us that we have got a white male rabbit here, or a hare. It is crossed with a black female, and all now the offspring are black. So what does that tell us? It tells us now that the black female has got the dominant allele, and then the white female has got the recessive alleles. Hence now the recessive alleles are masked in the phenotype of the F1 generation. The second diagram, which is pair B, they are just interchanged here, where now instead of the white male, you have got the black male and the white female. So now these ones are not influenced by sex. So this is not a sex-linked inheritance, but it just shows us that if you have got dominant alleles, they will be expressed in the phenotype, because they always mask the expression of the recessive alleles. Now, moving on. Now, let us look at another one of Mendel's laws. Now we have the law, or the principle of segregation. Now, what does the principle of segregation say? Now, the principle of segregation says that an organism possesses two factors that are now known as alleles, Now these factors separate or segregate so that each gamete only contains one of these factors. Now remember, in DNA and in meiosis, we have learned that during gamete formation, now a gamete, or rather a sex cell, will receive half of the DNA from the mother and half of the DNA from the father. Now remember, during meiosis and during metaphase, when you have got chromosomes lining up at the equator, and in anaphase they are separating, remember, DNA, we find it embedded in chromosomes. So when chromosomes separates, it means that that separates DNA. So during gamete formation, if here you have got an individual that is heterozygous for height, it means that now a gamete can only contain one of these alleles. So a gamete of this particular organism will either contain the allele that is dominant or the allele that is recessive, because now a gene, this one here is a gene. These ones here are alleles. Remember, we had said earlier that offspring do not inherit genes from parents, but they inherit alleles. So now these alleles, they separate during gamete formation, so that now the gamete can only receive one of them. So that is Mendel's law of segregation. We have now come to the end of our lesson. You can leave your comments. Don't forget to leave the name of your school so that we can be able to give a shout out. Please share, recommend, and subscribe to our YouTube channel. And thank you for watching.